This is a production of Cornell University.
So the hard part here is that it's really hard to imagine from the fly or beetle's point of view what it's like to be here. This is the scale of the building across the street for you and me, right? For a beetle or a fly, this is a high-rise tower. And so when they're in that chamber with all the flowers around them, and it's hot, and it might be a little narcoleptic because there's a lot of carbon dioxide in there, and so they might get a little sluggish, and they're bathed in odors that release behaviors that are reflexes for them. They're not even, optional. It's hard to imagine what it's like for them to be in there. What we do know is in all the trapped flowers, there's a rhythm. When the heat turns off, or the carbon dioxide turns off, or the scent changes, then they mobilize, they leave. And we don't know when they leave in this flower because we've been watching it in greenhouses instead of in Sumatra. So on D-Day, whether that's Monday or Tuesday or Wednesday, the spathe here will begin to pull away, okay? And what you'll see, you can just see that a little bit here. I don't want to do that too much, but it's flushing. Yeah, it's beginning to flush with pigment. Okay, when it's fully open, and you may have seen this on photos, it's a rich burgundy, almost into violet. It's quite beautiful to me, actually, and very, very textured. But that'll come away, that over maybe 6 hours on the final day, it'll sort of come away like a skirt. Okay, and then you'll be able, like the konjac plant, to look down into it. Okay, and you'll be able to see there's a high color contrast in there because the burgundy goes down and then it shifts completely to yellow. So there's this really strong visual contrast involved. There's also a lot of texture. All of these pleats and folds are not going to be lost. Okay, this becomes a funnel. So it's pretty remarkable. Something that I remind my students a lot is that even though we're focused very much on the chemical aspects of this, because that's a frontier in our field, this is a multi-modal experience for the animals as well as for us. Color, shape, texture, temperature, okay, ambient things like carbon dioxide and oxygen, as well as very, very strong and informative odors. So that'll come away. Paul, do you remember was it hot to your touch?
I think so, and
Ellen, yeah.
We recorded it something like about 100 degrees Fahrenheit.
Yeah, it's maybe 38 centigrade. It's at least 10 degrees centigrade above ambient, which is astonishing.
The core stays right there, just like that.
Yeah. This won't move or change. What is interesting and is very poorly understood is at the height of the thermogenic event of the heating, this will start to exude fluid. That's right. And it's not clear to us yet, this hasn't been well studied, whether that fluid is coming as a secretion from the cells on the surface of the cuticle here, or whether it's condensation because of the huge thermal gradient between the surface of this thing and the atmosphere around it. Either is possible. But what some people have done, and there's a Japanese study that was just published, actually collecting the fluid and looking at the chemistry of it, it's passively absorbing a lot of chemistry. So this is the tower stays as it is. The spathe becomes a skirt and it opens. And then you can see down into it. And then I think the males are on the bottom or on the top? What's the order?
Males are on the top, at least in contact are on the top.
Okay. Yeah, that's typically the way it is in aroids. That the female florets are on the bottom. The insects are often on the bottom with them, and then they have to walk out over the male. And that's a nice way of not getting your own pollen, on your own flowers, because it's not. Yeah, and so then you get to think about the plant compartmentalizing its reproductive biology. Here it's using carrion mimicry to affect pollination, but then fruit dispersal is kind of honest advertising to birds and mammals who are really interested in the color orange and red. It's very, very bright. We have photos on the website of how visually enticing these fruits are. If you've seen jack-in-the-pulpit fruits here in our forests, scale that up, as Paul says, like a medieval mace.
This has been a production of Cornell University, on the web at cornell.edu.